So I suppose we should get started. Wanted to remind everybody that our fourth lecture quiz is slated for Thursday, and we already mentioned, I think, that that was going to cover chapter 7, which is the microbial nutrition, ecology, and growth chapter, which you should be wrapping up as we speak. Any questions on that? It's been a while since we've taken a quiz. Whole week or two, maybe. We didn't have a quiz last week, did we? So seems like we were having something. We were quiz or something every week for a while there, weren't we? All right. Any questions on that? All right. How about questions related to chapter 6, viruses, or 7? I'll be starting to post the PowerPoint lectures for chapter 8 this week. I started looking at it actually, and it's kind of a long chapter, so we might maybe do three lectures or more rather than 2. Okay, floor is open. What would you like to ask about, go over, etc., etc.? Everybody's good with viruses? No questions on the virus chapter? I know we talked a little bit about that last week.
I have a question.
Okay.
So on the chapter 7 stuff, so it talks about like the heterotrophs and like the autotrophs. Could we like go over that a little bit?
Sure.
That section. Okay, thank you.
Let me pull up the PowerPoint. Okay, it should be pulling up in a second. Here we go. So the major difference between the two relates to the autotroph being able to, in essence, get its organic compounds from other sources, but typically not as a result of having to eat, let's say, or assimilate another organism. We are not autotrophic like, say, a plant would be. A plant can utilize the sun's energy to make organic compounds. In other words, the energy to do that comes from the sunshine or from artificial light sources. You can grow plants with grow lights, right? So you need light. So that's the energy that helps that plant or that photosynthetic bacterium. That's energy that's going to be utilized to help that cell make, say, other organic compounds, proteins, carbohydrates, lipids, and that kind of thing. It still needs a carbon source, and we as heterotrophs need a carbon source, but our carbon source comes from, again, assimilating is the term that you can use. Eating is another synonym for that. We eat. Bacteria don't eat, do they? Well, I guess they do. They don't have teeth, but they bring materials in. They have intracellular enzymes to break that material down. And that source of carbon is what the organism is going to use to make its own organic compounds. Enzymes is a good example of that. It's a protein. Without assimilating food, if you will, we would have a hard time surviving. We know we would starve to death. And the energy comes from the food that we eat. We break that down and we utilize it in a process called cellular respiration, which again, we're going to get into in chapter 8. We'll review that. But we make cellular energy. We make ATP by harnessing the energy that's stored in food. A plant, we just said a moment ago, utilizes outside energy sources, the sunshine, for example. Where does it get its carbon? We know we get our carbon from food we eat, but plants don't eat. I mean, there are some parasitic plants that eat, I guess, like Venus flytraps and sundews and pitcher plants, but that's kind of an exception to the rule. How does a plant get the carbon it needs to make more protein, more carbohydrate, more lipid, to grow, to produce fruit, and all that stuff? Do you remember this Bio 101 in high school? Where does a plant get its carbon from?
The soil, isn't it?
I'm sorry?
Is it the air we breathe? I don't know.
Well, you're getting really warm. You're getting really warm. We inhale oxygen, don't we?
Yeah, and don't we exhale carbon dioxide?
Right, and it's the CO2 that is the carbon source for plants. Exactly right. Yeah. Now the nitrogen and the hydrogen and other important elements. Remember chopsin? We talked about chopsin at the beginning of this this PowerPoint. Plants get a lot of these other materials via absorption through their root systems. Okay, and I know we're going off on a kind of a tangent here a little bit on plants, but when you think of autotrophs, that's what I often think of first off the bat is photosynthetic organisms. But algae are photosynthetic organisms too. They're microbes. We have cyanobacteria. They're prokaryotic. They can undergo photosynthesis. So in some sense, they're somewhat similar to plants in that ability to use sunshine. So that's just one of the more common ways that autotrophs get their energy from the sun. But we also talked in this chapter about chemoautotrophs. So here's an example in this slide. So photoautotrophs using the sunshine, like photosynthetic organisms, i.e. algae, so forth. Chemoautotrophs, they are getting their energy not from light, like algae do, but they're getting their energy from chemicals in their environment. And this is where I asked you to go back and look at that video that I posted in one of the earlier chapters of the deep ocean vent communities, these smokers. They're fascinating geologic structures at the very deepest parts of our oceans in the pitch black with very high pressure. And they were first discovered in the 1970s by these like two-man submersibles that went down and so their lights on, and they saw these smokers, which is basically energy being ejected from this Earth's core, right? There's no sun down here, black. But yet the bacteria that live here are getting their energy from the chemicals given off by our Earth. And they're converting that chemical material into usable forms of energy that allow them to survive. They're also getting all these other compounds, the chopsin elements, for example, that's also being provided to them from these emissions, if you will, from the core of the Earth. And we talked about how that provided a basic framework from which higher level organisms also live, like these big tube worms, these meter long tube worms that live there, and the blind shrimp. And remember that video that I had you watch? Fascinating. Go back and watch that again. Might make more sense now that you've heard about chemoautotrophic examples. So contrast the autotroph again, be it photo or chemo, with heterotrophic organisms of which we are members. We are chemoheterotrophs. We have to eat food. Most bacteria, for example, would be chemoheterotrophs. They get their food from absorbing outside sources, either through what you had for lunch, or bacteria would absorb organic compounds from their surroundings. And that not only supplies them with the necessarily elements to make organic compounds, but also the energy that's derived from breaking that down. Yeah, so that's what this kind of gets at. So most microorganisms, and we'll just say bacteria being the predominant example of that, would be chemoheterotrophic. But we did talk earlier in one of these chapters, and also in lab first or second week, we looked at some photoautotrophs too. Algae being the example that we talked about, but even some kinds of dinoflagellates and other eukaryotes, of course, can do that too. Does that help kind of clarify that a little bit? Great.
Yeah, that helps.
Yeah, it's a topic you probably heard about maybe in biology in high school, but we didn't spend a whole lot of time on it. Other questions on this section of chapter 7? Or we could again talk more about any of those topics. Like to review, go over things that aren't quite clicking. Are you finding the terminology difficult at all?
I have a question.
Okay.
Could you go over the equation for the rate of population growth?
Yeah, we're not going to get into the calculus of that, if that's what you're concerned about. But I will flip to that slide.
So do we need to know that for the test or anything?
No.
Oh, okay.
Let's see, where was that? That was in the later part of, yeah, here it is. So Gabby's referring to this equation, aren't you? Is it on your screen?
I can't see it.
Okay. Right here.
Yeah, that one.
So all this basically is telling you is if you know the initial number of cells, and if you have some idea of the generation time, which is defined as the time it takes for the population to double, then you can calculate reasonably accurately what the theoretical number of cells might be after that time has elapsed. But I'm not going to ask you to calculate anything on the test. But I think, again, it's worth understanding the concept. I think it's worth understanding how what could be a very safe food to eat could quickly become a very unsafe food to eat if left sitting on the picnic bench all day, given the fact that some of these bacteria can double, every half hour or even quicker. And if you had a fairly high microbial load to begin with, let's say one that was acceptable, in other words, you could drink it or eat it and it would be fine, but it was on the very edge of going bad, if you will, and you provide it with just really super great growing conditions, boy, you could get sick eating that egg salad sandwich, theoretically. And this also brings up a sort of another side topic, and I don't want to take up time if you have questions too, but when we talk about, buying milk, we have a date stamped on that gallon of milk, right? Like today's, what, the 9th? So if you bought a gallon of milk today, you would probably have an expiration date, I'm going to just take a wild guess, somewhere toward the end of the month, right? Probably 2 or 3 weeks. And I was just reading something in the paper about food wastage, how much we waste food in this world we live in. It's like unbelievable amounts of food gets dumped, tossed because it goes bad. Some of this occurs because people don't eat their leftovers. They rather just throw them out. But oftentimes they do go bad. But that gallon of milk, when you buy it, that's not sterile. You all understand that. There's an acceptable microbial count in that gallon of milk that you buy at Reed's or Tops or Wegmans or wherever you go. But by the time you get to March 27th, does that mean that after March 27th, you should dump it down the sink? What do you think? There are people that believe that and will do that. You do understand this, don't you?
Isn't that like the best buy date? Or I don't know. I don't drink milk, so I don't know. But
Well, you bring up a good point, and that is, should we be labeling our food with not just the date, but a best buy date or freshest if used before date? You know what I'm saying? In other words, if you open that gallon of milk or use it on the 28th, if you've been keeping it refrigerated, odds are it's going to be fine to drink. You're not going to get sick just because it's the day after the expiration that was stamped on it. You all kind of know that, I hope. But it's just interesting, I think, that some people tend to not understand, I think, what that really means sometimes.
I know there's expiration dates on like water bottles, but apparently that's like the water bottle, not the actual water that expires. I've seen something like that. Like the people will throw away the water when the water's fine. It's just the bottle or something like that. And I was like, okay.
I wonder, are they saying that like chemicals leach out of the plastic, or what does that mean?
I don't know.
Jocelyn's shaking her head.
It's usually the container's no longer good, like it'll start to break down, so it can open it up to bacteria to get in. They'll do it a lot with a lot of medical supplies too. So like that breaks down and you don't see the hole or something, it's no longer sterile on the inside.
Yeah. So when you buy bottled water, people probably don't think much about the fact that they're ingesting not only water, but maybe some of the plastic chemicals that I don't know. I don't buy bottled water, so I don't know, but a lot of people do. That's kind of scary to think about it. What are you really ingesting sometimes? But anyway. So Gabby, you don't need to know the calculation, the equation. How about other questions on chapter 7? Anything?
Are we gonna need to know the different ways to analyze and count the cultures?
Specifically?
On page 220, it starts going into different techniques into counting or separating. Are we gonna need to know like the specific methods?
I think you should understand the concept of turbidity and population size change. And that's going to be super evident in lab next week, because we're going to do a lab that specifically marries the standard plate count or viable plate count method with what the spectrophotometer, how that's used in the turbinometry way of estimating population. So if you have lab, this is the big advantage of taking lab and lecture together, because it's going to be a heck of a lot easier to understand, because you're going to be working on that and doing that after next Tuesday. Now, some of you I know are not in lab, right? You've either taken it already or you're going to take it. I forget what the situation is. Some of you have already had it. Who's already had it?
I have already had it.
Okay. Anybody else already had it? Who has not taken it yet then? Oh, okay. So all of the rest of you guys are in lab or have had it. Okay, so that's going to help clear this up a lot. But get back to your question, do you need to know the various techniques? Yeah, I think it's helpful to understand, and let me just again go to that slide. I think it's helpful to understand that there are these techniques that are a bit dated. For example, can you all see the slide? So I talk in the PowerPoint lecture about this. I don't know how many of you had me for ANP1. I can't remember. Some of you have. Okay. When I teach ANP1, when we do the blood studies, I often bring out a slide and I show the students. It's called the hemocytometer slide. And on that slide is a set of grids that are etched into the glass of the slide. And what they used to use this slide for was to estimate the number of white cells and red cells in your body. Now, today, it's totally automated. It's very different. But back in the 1950s, let's say, and I know you're like, why are we talking about that? Well, because it's interesting to know how it was done in the olden days, so you understand that you can use the same sort of technique to do this for microbial numbers. What they would do is they would take a blood sample from a patient. They would use a special little pipette. They would draw in a small quantity of blood. All you do is prick your finger and draw up less than 1 ml of blood. You would dilute that with a Ringer solution, a saline solution, isotonic saline solution. And then you would dispense that drop onto a slide called a hemocytometer, not unlike what you're seeing on the slide. And then you would look at this under the microscope and you would see the number of red blood cells or white blood cells, depending upon how it was diluted and how it was stained and some other things. And then you would multiply those cell numbers, let's say RBCs. You multiply the number of cells you'd count within a particular part of that grid by a dilution factor, and that would tell you approximately the RBC count per cubic millimeter in the patient. And if it was done accurately, and a lot of lab techs did this 24/7, so they were darn good at what they did, they got really good accurate numbers. So you could tell whether somebody was anemic or low RBC count or what have you. And that's how they used to do it. And in a similar vein, that's how they used to estimate microbial populations using this direct cell count technique. The problem with using this is, as it says here, you're counting not only live cells, but dead cells. Do you think you can tell a live cell from a dead E. coli from a live E. coli under the microscope? No, you probably can't. So it probably wasn't the most accurate in the sense of you're not sure what percent could have been dead, what percent could have been live, blah, blah, blah. It was manual, obviously, it took a lot of time, and you had to have your techniques down to get good numbers. But nonetheless, that was a technique that was used. If you were a dairy farmer and you were getting graded based upon the microbial content in your sample, you were getting picked up every twice a week by the dairy that you were a cooperative member of, you were incentivized to keep your dairy as clean as you could, so you had a lower bacterial count, so you could make more money, right? And this is how they used to do that. They would determine bacterial counts in milk in using this method many years ago. So anyway, that's one way of doing it. Today, of course, it's high tech, and this particular flow cytometer system is just beyond my understanding in terms of the physics and the complexity of this. But basically, what they do is you can run a microbial sample through this machine, and you have to usually stain or treat the cells in some way. Here they're talking about using a set of different fluorescent dyes. And then using a laser, running a laser light through the sample as it's fed through a very small opening, the system will be able to distinguish between living cells and dead cells, and to some extent can even differentiate E. coli from, say, staph. In other words, bacilli from cocci. So this is super high tech. And then this graph showing, of course, Staph aureus or E. coli, they're giving you a graphical representation of the number of dead cells and the number of live cells. So you get a sense kind of quantitatively of what you might be dealing with in that particular sample. So to the extent to which you should understand, the basic system, then I think, yes, it would be helpful to know how basically a flow cytometer works. But just have a conceptual understanding of how it works. Notice that we not only can use this to analyze microbial, bacterial, things, we can also use it now to count white cells, red cells. We can probably even do a differential white cell count, where you're differentiating the percentage of neutrophils, eosinophils, basophils, neutrophils, monocytes, etc. That used to be all hand done years and years ago. Now it's all automated. So it's high tech, right? High tech. Back to the spectrophotometer and the turbinometry idea here is that if you look at a liquid sample, a broth, that's cloudy or turbid, that is an indication you have cells, right, that are preventing you from seeing through this. Y'all know that. You've seen that in lab. When you get that sterile broth from Deb, after she pulls it out of the autoclave, it better be clear. And it is, right? So the idea here is if you stick a broth sample, a liquid sample, into a machine called a spectrophotometer, this machine will send light through this column of liquid, not lasers like the flow cytometer did. This is just plain old light, which you have to regulate, or you have to adjust and calibrate the system. So it uses a particular wavelength of visible light. But you basically look at how much light passes through the sample, or does not pass through the sample. If it's a sterile sample, like we have, say here, which is corresponding to this test tube, you're going to get the majority of light passing through and detected on the other side of the light source. There's a little photodetector over here, right? Now, will any of this light get refracted off to the side by the glass of the tube? Yes, some will, but that's all taken into account. But look at this more turbid sample. Now, is some light going to get through and detected by the detector on the other side? Well, depends how turbid it is, but if it's slightly turbid, then some light's going to get through. If it's extremely turbid, there'll be a point at which there's so many cells, maybe light can't get through, but generally some light gets through. But the blue arrows you're seeing here are simply referring to how light gets diffracted and refracted off to the side when it hits cells, basically. In other words, less is passed through the tube, more gets scattered because of the cells inside the tube. Makes sense, just think about it. So what we're going to be doing in lab next week is we're going to be looking at various dilutions of broths that have cells in them. The more turbid, the less light that passes through. The more diluted the sample, the more light passes through. And we're going to connect the dilution factor with this idea that we can also not only can we pass the light through a diluted sample, a broth sample, but we could also take that same broth sample and plate it onto a sterile agar plate, incubate that, get a CFU number value. You know what CFUs are? What are CFUs?
Colony forming units.
Right. And you multiply the number of CFUs by the dilution factor, and you can come up with a number, an estimate of the population, right? So let's say that this is a particular dilution of an original culture, and we get an estimate of 20,000 cells. Well, we can also shine a light through that using the spectrophotometer and get an absorbance value, and then graph the absorbance value with the population and generate basically a curve. So that if we wanted to get an idea of how many cells were in a given dilution, we've already done the groundwork here, the grunt work, if you will, by plating all this out. Now we just stick the tube into the spectrophotometer, read off the absorbance value, and then go to our graph, find the absorbance value, see where it hits the curve, and then extrapolate down the population size. This is clear as mud, isn't it? Well, it's going to make more sense when you do it in lab next week. So when you get to that exercise, and I told the morning group, next week we're doing exercises 17 and 19. Spend some time on exercise 19 because that's what this talks about, the viable plate count or standard plate count, and also how the spectrophotometer is going to be used to help us better understand the relationship between turbidity and population size. But there were only just a couple different, examples that the book gave as to how to assess population, right? The slide, the flow cytometer. We're going to talk more about this coming up next week in lab. I don't know, Samantha, did that help? Okay. This is really important stuff here, the growth curve. The three different components, or actually 4 different phases here. Yeah, this is what goes on when you get your sterile broth in lab and you inoculate it. This is what's going on in that test tube. Now, again, this is kind of theoretical. These numbers don't mean a whole heck of a lot, other than you see it growing soon after the lag phase, and it really starts to grow fast as the cells are doubling in number each generation time. Yeah, this is pretty self-explanatory, I think, though, but interesting stuff. The chemostat, this is interesting as well. I talked a little bit about when you would use a chemostat. I'm not sure if you've gotten to this or not, but check it out. Very interesting. How about other questions? Are you finding chapter 7 not too bad? No? There is quite a bit of stuff here that I have a review stamped on the slide, I know, because we've talked about osmosis and diffusion and hyper isotonic solutions back in A&P, active transport, facilitated diffusion. If there's any of that you'd like to review, we could certainly talk about that as well, because I, again, did not spend time in the lecture going over those topics. Not that we couldn't. Any questions on 7?
I have another question.
Okay.
So the leaf cutter ants, so is that, I don't know how to say it, amensalism and not mutualism? I don't know what amensalism is.
Okay, so what 2 organisms are you wanting to talk about? The leaf cutter would be one. What would be the other organism? I mean, that's really what that gets at the heart of what sort of relationship you want to describe.
I just wasn't sure because in the book it was like, I don't know what it was.
What is it? What did we say?
Because I don't know if we talked about amensalism or not in the lecture.
Yeah, right. I think I threw this in here. Well, I mean, you could think of it as commensalism, I guess, in the sense that the ants benefit, but the fungus that they eat and grow isn't so much hurt or helped in the process. Although you could think of it as mutualism too, because the fungus is maintained and it grows and its progeny is carried on. But in that example, I also talked about parasitism as it related to the little minim cast here that, is riding shotgun to prevent parasitism of his brother down below here. And it gets to this whole idea that in the tropics, and especially in like insects like this, there is sort of gray lines depending upon all these interconnected food webs and relationships and so on. I think I threw that in just because I've seen it so many times when I go to Costa Rica and Panama. These are so common. They'd be like, we take robins for granted when we step out the side in June. You know what I'm saying? They're just all over the place. We don't think about them. They're there. We know they're there. We know what they are. And in the tropics, these ants are all over the place. They're always there. But to somebody who has never seen this before, it's absolutely phenomenal to understand that they have such a complex social biology, that they all have, these different caste systems or caste system that involves, some of these going up and biting these pieces of leaves off the plant, bringing them back to the subterranean nests. Others are guarding the workers here. It's just a fascinating example, I guess, of a symbiosis. But as far as, is it commensalism? Is it mutualism? I guess it depends on how you look at it. If we're talking about the ant and the plant, well, then I guess I would kind of call it parasitism. The plant's getting harmed. Not that generally these ants denude the plant and kill it. That's typically not what happens. Because there's so much productivity in the tropics that a plant that's denuded, like this picture that I showed you earlier, you might think this plant's going to die. Well, it's probably not going to die because within a few days, there's going to be new shoots coming out, and it might even be growing faster than before it got influenced by the ants. But I mean, the plant's getting harmed, it's getting eaten, so it's parasitism, I guess, in that sense, right? The fungus that the ants are growing, I don't know, is the fungus harmed? Well, it's getting eaten, so I guess if you're being eaten, you're being harmed, but you're also being cultivated by these ants. So is that bad? You know what I'm saying? It's sort of philosophical in some kind of weird sense too. The point again is that it's an amazing relationship that these organisms have with other organisms. Yeah. If you ever go to the tropics, you'll see this. Has anybody ever traveled to the tropical areas? Anybody? No? Maybe you should go sometime.
I've been to Punta Cana.
Where's
So
that?
that's the Dominican Republic.
Okay.
So that's as far as I've gone.
Well, I suspect they have some pretty interesting organisms there too. I'm sure they do. They probably
Oh
have
yeah,
kind of-
it's crazy there.
Yeah. The section here in biofilms is really, really important. Really important. And have you had a chance to look at this TED Talk yet by Bonnie Bassler from Princeton? You should be looking at that. That's fascinating. This video actually that she did, it could be 8 years old now. I'm not even remembering what year she did it, but it's still very relevant, very interesting. And for those of you going into nursing, which is the vast majority of you, this second video you're going to find really cool. It talks about the role that these have on chronic disease.
That's one of like the big things. Like people have like a urinary catheter, and so
Yeah.
like every single time, like every day, you should be like talking to the provider and making sure that catheter is still like a relevant thing. Like, do I need it? Because the longer it sits in there, like the urine's like staying still, and the longer that's like that, more bacteria grows.
Yeah, exactly right.
Biofilms.
Absolutely. I don't think that now maybe I'm speaking out of turn. You tell me, nursing people, if you've heard this in your nursing classes. Do they talk about this much, biofilms, in any of your nursing classes? They probably do a cursory mention of the importance of changing catheters and endotracheal tubes and all that stuff.
Yeah, I don't think they meant like call it biofilms, but they say the bacteria like builds up.
Yeah, but
And
I think,
yeah.
right. I think as a nursing student or as a science student, you should have a good understanding of this because number one, it's just really interesting. And I think it holds the key. If we understand how biofilms work and how these cells communicate with one another, as the Bonnie Bassler TED Talk will talk about, we can use that knowledge to counteract the microbe. In other words, treat the disease in a totally different way that was unheard of or not even known about not that many years ago. You know, we think we're so advanced, but in another 100 years, people are going to look back on people that lived in 2021 and say, what sort of cavemen were they? Now, it's interesting, but you go back to 1921 and compare where we are now versus then, we'd say the same thing. They were living in the dark ages. They didn't even know a heck of a lot about surgical techniques, or they understood, you know, the germ theory of disease a little bit, but you know what I'm saying? So every decade that goes by, we learn so much more, and we're never going to be done learning. You sometimes think you reach a point of, well, I can't learn much more about this. Well, yes, you can. We can learn a heck of a lot more about lots of things. So I think, you know, being open to change and new things is tough in the medical field. We are resistant to change. Nobody really often embraces change. Nobody liked, you know, what Lister said. Nobody believed what Semmelweis said. Some of these folks we stand in awe of historically today. They met with massive resistance, and they had enough guts to stand up and speak their mind. And then years and years and years later, after they've died even, we come to understand that, you know, these people knew what they were talking about. We should have given them more credit and listened to what they said. And many lives could have been saved maybe if we had done so. And so just kind of always be thinking, don't get stuck in a rut. Always be open to new ideas. Yeah, biofilms, really neat stuff. Yeah, we did not use the term Mentalism like the book does as it relates to the leaf cutters, but that technically is what it is. But I did not specifically use that term, so I wouldn't worry about it. Okay, anything on chapter 7, chapter 8? Okay, well, look for some videos this week on chapter 9, at least the first part of it. We're also into that chapter next week. So this chapter, again, a lot of it is somewhat of a review, or should be a review. But this gets to be a little bit technical. This is the chapter that talks about enzymes, how they work. It talks about metabolism. It gets into cellular respiratory processes like glycolysis and Krebs cycle and electron transport. Only it will, again, go into some specifics as it relates to, say, aerobic or anaerobic bacteria. So I guess what I'm saying is this chapter may be a little bit challenging, and that's okay. But a lot of this we touch upon in A&P 1. I tend to spend more time on it than most instructors do. So if you had me for A&P, you probably are maybe at a slightly better advantage in terms of, I think a lot of instructors just don't spend much time on this, and they should. Okay, well, I will stay in the Zoom room until my micro lab this afternoon. So feel free to hang around, ask questions, chat, whatever. Otherwise, I will see some of you in a half hour or so and the rest next week. And as I said, we'll have that quiz, that 5-point lecture quiz 4, ready to roll on Thursday. Some of you are taking it during that normal 1:15 to 2:30 time, and others who you know who you are. I know who you are with respect to special other times. Okay, great. See you shortly.